Okay, welcome all to the last Microbial Cell Factories webinar series. So my name is Erika Takano. I think I've met some of you who have come before. And this is organized by ourselves, myself, through the TOPCAPI project, and also with Professor John Morrissey, who is also attending today through his project in CHASSY. So this 3-webinar series, we've been talking about microbial cell factory development from concept to commercial development at all stages in between. And if some of you have joined us before, the first talk was on in silico design and programming of microbial strains. The second was on strain engineering approaches to build cell factories. And so today, which is our last webinar series, is on validation and about compound analysis and transferring technology to commercial scale. And we have 3 excellent speakers who will be speaking today. And the first keynote speaker is Dr. Sophie Roelants from the Biobase Europe pilot plant in Belgium. So Sophie, perhaps you could start your presentation. I will stop sharing.
Okay, hope it works. Share screen. All right. You should see it now, I hope.
Yes, if you can make it full screen, perhaps that would be good.
Is it working like this? Okay,
yes,
perfect.
Perfect, thank you.
Okay, so then I will kick off. So I would like to start by thanking you for the invitation. It's nice how you set this up, and I hope there will be some take-home messages that people at the early stage can benefit from it, that we can all make it work faster and better. So hi to all, I'm Sophie Roelants. I work at the Biobase Europe pilot plant, but I also work at the University of Ghent, both in Ghent in Belgium. And I will hope to convince you with some tricks to consider scale up early on. So we'll first give you a brief explanation on what the Biobase Europe pilot plant is and what we do with scaling, so that you know a bit the context. Then I think the most important part is to provide you with some insights on our experiences the past 10 years, on what you should consider and when you should consider it when you're working in this field. And then I will also give you my personal approach and a case study on a topic I've been working on for over 10 years now. And that will link them back to the tips and tricks. So I've gone through all these things that didn't go well or that we didn't consider early on. And I hope that I can convince you in the early stage to do so. So very shortly, Biobase Europe pilot plant is a multi-purpose pilot facility in Ghent. So we work on any type of bio-based products and processes. And as such, we want to enable and accelerate the transition into a bio-based economy. So we do this by process development and scale up. We were founded a little bit more than 10 years ago. We are independent, so we are not linked to any big industrial shareholders, which means that your secrets are safe with us. It's a very important aspect. And we're now a little bit over 100 employees. When I started working there in 2015, we were 30. So it's a fast growing company. So we see that this is really happening. Bio-based economy is growing and people are coming to us for that, to help them. So what is our role then? So I think a lot of people, I don't know if you can see do you see my cursor when I move on the screen? Maybe you can do it like this. Yeah, like this.
We see it now.
Yes, okay. We see it now.
Yes. So typically, I think in the previous webinars, we talked about more, not fundamental, but mostly research at lab scale. And at the other side of the spectrum, you have the commercialization of new products. In between, you have what we call the valley of death. Typically, technological risk for demonstration of a new process is still quite high, while the capital requirements also rise. So it becomes quite difficult for people to overcome this phase. And that's where we come in. So we typically say that we close the gap in the innovation chain. As bio-based Europe pilot plant, we actually work on a lot of different aspects. So we can work on very crude biomass, for example, side products from agriculture. We can work on
Oops, maybe we've lost Sophie for a bit. Sophie,
We've
are you
lost her?
back? You're
Yes.
back. Yes, good.
Oh,
Can
did I?
you
Oh,
carry on?
sorry.
Yeah. No, it's all
Yeah,
right.
I don't know where I lost you, but so we typically work on three types of unit operations to convert biomass into refined products by also applying purification, so downstream processing. To show you a bit how the company looks like, so it was an old fire station which is refurbished into a pilot facility. So the heart of our facility is our lab, where we do all our analysis and also our process development. Then we have a process hall for pre-treatment, which is here. We have another process hall for fermentation, where we have about 15 fermentation vessels of 30 liter until 15,000 liter scale. Then we have a green process hall, which is here, where we can work with solvents. Typically, we use that hall for solvent-based downstream processing, purification. And then we built a few years ago a new process hall, the biggest one actually, for water-based downstream processing and, for example, spray drying. So to really get to the refined dry product. So we convert biomass into refined products through using several processes. And here you can see some examples of products and things we worked on in the past. So it goes quite broad. And my specific topic that I work on are biosurfactants. So that's also the case study that I will present in the last part of my presentation. Then just to say typically how we work in which type of projects. So we are currently involved in 20 consortium-based projects, which you might all know, typically Horizon 2020, BBI, and then also some national projects. But besides that, we also work with companies on a bilateral base. So last year we had about 80 bilateral projects with 32 customers. So you see some customers do more than one project with us. And we also see that customers come back over the years. So that was the brief introduction to the Biobase Europe pilot plant. Then I will move to the important part, I think, to the tips and tricks. So you see some quotes here, which I might also have to plead guilty for in the past, that I didn't think about certain things that you think, oh, these are things to consider later on. It's not important to me. But I hope I can convince you that it's not entirely true, and I will give you some tips and tricks. So you should start thinking about the scale up and what comes after the lab scale as soon as possible, so early on. So an important thing to consider is, are you developing a bulk chemical or are you developing a specialty chemical? Why is this important? Because the production cost and the cost of your final product will vary for these types of chemicals, which also means that you might, in your development, have to focus on different aspects. For example, substrate costs for a specialty chemical might not be so important, but for a bulk chemical, which can be in the range of €1 per kilogram, that you can sell it on the market, this needs to be quite low. So you cannot afford to use very expensive compounds in your process. And these are things that you typically find out by doing a techno-economic analysis, which I also would advise you to do early on, as soon as possible. Because we found out, for example, in our process, we thought we had to focus on medium to get it cheaper, but actually the productivity was a lot more important, and the substrate cost at the lower scale was not of any importance into our production cost of our final chemical. Also, when you start developing a process, think about the scalability thereof. We often get people coming to us with really fancy things they developed in the lab, but that will never make it a big scale, that it's too difficult, too complicated. That also relates to this tip, please keep it simple. The more complex you make it, it might seem very innovative and nice, but if it's too complex, then it will also typically not be scalable and you will have to start all over again. A second thing that I also personally encountered multiple times in my career thus far is the importance of analytics. It's crucial aspect actually in every phase of development, research, demonstration, and also commercial phase. So you need to know what you're producing and how much. So you need ultra pure standards. If you cannot buy these standards, with Sigma Aldrich, for example, you should consider making them yourself. Because if you cannot quantify your product, you can also not follow up your process and you can also not optimize it, or it becomes quite difficult. There are some sensors that have been developed lab scale, but typically when you go to scale, this becomes difficult. So please try to consider this as early as possible. Also, it's important to know if there are any byproducts produced. Also, beware that you might not see a byproduct in the method you use to quantify your biochemical of interest. And a very important one, which I bumped into quite a lot already, is that you often think that you get the full picture, but it's not because you don't see something that it's not there. I know that's a difficult one, but it's just the mindset to allow that. In research, of course, a lot of things are not what they seem, and especially related to analytics, we've seen this quite sometimes that we publish something or we file a patent, and then afterwards we actually find out that it's completely something else or different, where it was actually due to the limit in our analytics. So an important one as well. Then optimize your fermentation medium. So also, this is something to consider early on. Don't think this will come at a later stage. It's important to consider this early on because it's also linked with your strain. Doing techno-economical or a life cycle analysis will learn you where you should focus your activities, your efforts. Is it the productivity that is most important? Is it the yield? Is it the titer? Can be even something else. So when looking at the fermentation medium, you should know what you should optimize for. Also try to minimize the complexity of your medium. We often see people coming to us with very highly complex fermentation media that they develop lab scale. If your medium requires us to mix over 10 compounds and we have to add some through filter sterilization, others have to be autoclaved, it becomes really very complicated. It adds to the cost of your process, and also it adds to the fact that it can fail. So the simpler here as well, the better. Also, when you start using lab scale components in a medium that you can buy, again, as Sigma Aldrich, for example, we often see people coming to us with media that contain compounds that are just not available large scale or that are extremely expensive. And then they have to start all over again because it's just not feasible to scale up with those specific compounds. So that's also something to consider early on. Then also related to that, often in research, people use antibiotics or oxotrophic components as a marker, but if you have to keep on applying your selection pressure in big scale, that becomes really expensive, especially linked to antibiotics. Adding kilograms of antibiotics or tons of antibiotics to a fermentation is extremely expensive, and also if you have to dispose of your wastewater afterwards, that will also be extremely expensive to guarantee that you're not dumping any antibiotics into the environment. So it's always better to go for genomic integration, that you don't need to apply the selection pressure the entire time. And if you really have to use a plasmid, then try to place an essential gene on the plasmid together with your production pathway or gene, instead of an oxytrophic or an antibiotic marker, that you don't need to add specific expensive compounds to your medium. Then another one that is important is time is money. So the faster your process, the cheaper your biochemical will be. Consider that in a typical process, still the operational costs of the people are the most expensive factor. So, and also, yeah, you using the equipment, taking it, and it cannot be used for another process. Also, the slower the process, the more the chance of bacterial contamination. And here also beware that a lab condition where it's really quite easy to work in sterile conditions, that it's not the same as an industrial scale condition where contamination is a lot more difficult to control. So here as well, if your process goes faster, it will be cheaper, but it will also be less prone to contamination, so it will be more robust. Then also another one that we often see is that people come to us with a fermentation process, but they didn't really consider the development of a good downstream processing method, which is actually crucial. It's good to do that early on. It can also help you to make these ultra pure standards, which I mentioned in my first point, the analytics. Typically, we sometimes see people coming to us, yeah, but we have a HISTAC purification method that's typically not economically scalable unless the thing you're purifying is extremely valuable and expensive. So try to think outside the box and to think about novel approaches. We also often get solvent-based downstream processing. It's not a problem. We can do it. Big scale, it's also feasible. But yeah, in the context of a bio-based economy, try to think about the sustainability of your process, and also using solvents makes your process and your product more expensive. And last but not least, yeah, no downstream processing, no product. So it's quite crucial. So also something to consider early on. And then, last point that I wanted to make is that scale up is still part of the development phase. So it actually should be closely interlinked and intertwined with R&D research lab scale. That's also what I will show in my case study. It can be very slow and painful. The messages that I brought earlier, you might have to restart all over again if you didn't consider some points early on. Expect the unexpected. Scaling up is not always straightforward and linear. You can see some really unexpected things. So the work is not ready. Tech transfer is key. So you might have a lot of things in your head, little things you do when you do the process that are not in your protocol. So when you start writing protocols, try to include everything, also the small details that you think that are not important. They might be crucial for us to repeat your process at scale. And then, yeah, to add them all up again, and just maybe if you don't succeed, maybe you can look back at the tips and tricks. I actually made quite some errors or things in the past that went wrong, and I actually can find them back here, so maybe can help you. And then I will give you, will guide you briefly through a case study I've been working on for the past 10 years. So we work with novel types of microbial biosurfactants, which we actually want to bring to the market, and yeah, where we are actually now going to commercial scale. But we started really small lab scale about 15 years ago. That was done at the University of Ghent in the research group IMBio. And then a few years later, we started working with the Biobase Europe pilot plant for the process development and scale up. And actually what we've learned, and what also comes back in all these tips and tricks, is that applying an integrated design where you actually already start thinking about process development and scale up early on, and have an iteration with your genetic manipulation phase, it really helps. It really helps you to ask the right questions at the right moment. So we did that in several national and international funded projects. over the past years. You can look them up if you're interested. And then just to give you some background, so surfactants and so also biosurfactants are actually applied in a multitude of sectors and applications. So you can see some examples here. You can imagine it's quite a big market, so 20 million tons produced each year. And as you can see, still a big part is derived from fossil resources. Another part is derived from oleochemicals, such as palm oil, of which you also know is associated with some issues. And then you have 4% that is completely bio-based, and that's what we just work on. Completely bio-based, you can make it through chemical production. You can also apply biological production methods, and we then use fermentation. So we call these the microbial biosurfactants. They have some clear advantages, which is why we're interested in them. But obviously, they also have some disadvantages, and that's what we focused on to bring novel products to the market. So specifically, they're more expensive. There is not a lot of variability, and mixtures of molecules are produced. We'll speed it up a bit here. So what we did is we applied this integrated process design to really, in the end, bring products to the market. And we did that by genetic engineering. So genetic engineering of a yeast to really increase variety. So you can see a portfolio of biosurfactant compounds that we were able to produce. And here we really needed this analytics that I mentioned in the beginning. If we didn't focus on analytics really early on, we couldn't have gotten to the phase where we could analyze and detect all these different and new types of molecules. So that was extremely important. So I will then focus on one type, lipids, so a novel type of biosurfactant that we developed. We saw when we developed the strain at university first that the productivity was quite low compared to a wild type strain that produces another type of biosurfactant. So we said, okay, from the pilot plant, hey, university, you should do better. So they did some strain improvements, which already helped us quite a lot. And then we also started working on the process at the pilot plant. So we said, okay, let's focus on the fermentation. And as you can see, we went from a productivity which was quite low in the beginning to a productivity which was at least 10 times higher. And we saw in our techno-economic analysis that this productivity was actually key to lower the production costs. Purification, another one that I mentioned, very important. In the beginning, we reached really low purities, which we were able to maximize to above 95%. And also the recovery. Here you see 35% recovery, which means that we were losing 65% of our product in our purification. So that means that your product will become a lot more expensive because you're losing more than half of it. And you see by focusing here, we could also have a really high, nice recovery. So then we went to scale. The scale-up provides us data to perform techno-economic analysis, LCA analysis, and also the sensitivity analysis to provide us with the hotspots in our process to focus on. I quickly show you this graph. So we did this at 15 cube scale, and you saw that we still reached a productivity which was quite high for a first time. So we were quite pleased with this. And then we were able to produce a techno-economic analysis to actually anticipate how cheap we could get the product at bigger scale. At a productivity of 1 grams per liter per hour, you can see we still are at 20 euros per kilogram, which is quite high for a bulk chemical, which should be around 1. So you see, we still need to focus on this productivity. This is very important, and this was what we found out in this sensitivity analysis. Another point was that we could make, by scaling up, we could make kilograms of product, which we could test in a whole range of sectors because it was a new product. We didn't know where we should use it. Some application tests were performed for detergent application, typical parameters that were evaluated. We got some nice scores, but we saw that the stability of the product was not good. So we were already at 10 cube scale productivity, stability was not good. Because there's an ester functionality present in this compound. So we said, okay, University of Ghent, you should improve this. You should get glycosidic linkage here instead of this ester linkage. This will make our product a lot more stable. And so they did. They made a new strain that produces bola sophrosides instead of bola sophrolipids with 2 glycosidic linkages instead of this ester linkage. And indeed, the stability was dramatically improved. So then this company was actually quite pleased, and we are now looking, not specifically with this one, but also with others, to now bring those products to the market. So I hope that I have convinced you that by applying this integrated design, very shortly explained, that we can actually bring new biosurfactants to the market by applying this integrated bioprocess design. And still, of course, we have some things, some hurdles to overcome, but yeah, we're getting there. And that was actually it. So to sum it up again, and then I would like to thank you for your attention. I'm sorry I went a little bit over time, but I hope it was useful and interesting. Thanks a lot for your attention.
Thank you. Thanks very much, Sophie. It was a very nice overview. Let's see, let me just look. Maybe I can start with asking some questions, because I found your company very interesting, because you're doing everything, kind of all different compounds, all different, from fine chemistry to like surfactants, and also lots of different microbes, I guess you're using as well. I guess you're using yeast, but maybe plant, I don't know, maybe plants as well. Are you using plants? No. Just microbes, but yeast.
and enzymes. No, typically people come to us with all types of microorganisms. So now, okay. I gave you the example of the yeast for the biosurfactants, but we typically use any type of microorganism, if it's not pathogenic, of course, that the company or university might want us to work on.
Yeah. So my question is there some specific species that are really difficult to use that you would kind of go, okay, hold on a second, this is not really well defined. Yeah. We don't know what it is very much. We have no idea about growth conditions and things like this. Do you turn some of them down in that sense?
Yeah, we have some on the blacklist indeed. So of course, pathogens, we don't want to work with those, or opportunistic pathogens. But another one that is very problematic for us as a pilot facility are spore formers, such as bacillus. Bacillus, of course, it's a very interesting organism and can do a lot of useful things with it. But yeah, when we start growing those and they start making spores, then we see in subsequent fermentations with other microorganisms that we might bump into issues. We're obviously looking also into maybe having a separate space in our facility where we can work with those, because they are interesting. It's not because we, as an open facility that works with a lot of different strains, have difficulties with it, that it's not viable to go to big industrial scale. But that for us is an issue.
When you say, sorry, Sophie, when you say they sporulate, you mean they sporulate in liquid?
Yes.
I guess within your fermentation tanks.
And then they are very resistant spores, so it's really difficult to then kill them off. And we see in subsequent processes that we then often run into issues.
Oh, okay.
But yeah, it's not, I'm not talking against bacillus, because there are some really nice industrial processes out there that use bacillus. But for us as a facility that works with a lot of organisms, it's an issue.
Right. I guess. Thank you. Thanks. John, perhaps you have some
I have a couple of questions, but actually I see one in the Q&A first. Maybe we should have reminded people, if you have questions, you can type them in the Q&A, and Rico and I will read them out. But I see the first question I can see here, Sophie is asking whether Rhodotorula tyroides is on your blacklist.
It's not, no.
I didn't think so.
It'll be interesting to hear what's on your blacklist, actually.
And then there's a question in a similar vein. I think it's around bacillus. And it says, regarding bacillus subtilis or lecniformis, etc., would you take a project on if the spores were already less resistant, or if the strain is genetically engineered? I'm not sure what the genetically engineered bit means, but if I guess the question is, if the spores aren't particularly resistant, does that mean it's okay.
If that is proven, and if we can see the test that it's true, yes. And we've done projects with bacillus in the past, so it's not that we don't do that. What we don't do anymore is to specifically make spores with bacillus. Sometimes people want us to really make the spores, then you're really making your life difficult. So and the person that asked that question, it would be very welcomed, I think, to work with strains that have less resistant spores. Yes.
And one kind of, I suppose, question or maybe comment or observation I had, just based on your case study when you were describing at the end there, I suppose from most people who are interested in strain development, I'm thinking of maybe in the research labs in university settings, don't have access to something like the pilot plant early on, and but maybe they have access to bioreactors. And would you be saying, because ordinarily we do, as you kind of described, you try and get your strain pretty good before you start doing anything in the bioreactors. Are you advocating something different? I mean, are you saying, look, once you got your strain, I'd say almost producing anything at all, to really start even with what's really going to be a suboptimal, substandard strain, to start trying to develop the bioprocess with that?
At a certain level, it makes sense, because especially when you're making novel compounds, it's often difficult to know if it's valuable or where it's valuable or where you should focus on. So making the first grams of the product to give to interested parties, that is very crucial and important, especially if it's not a drop-in. If it's a drop-in, that's less of an issue because you know what you want to produce. And indeed, I'm advocating for a more integrated approach where you don't say, okay, now we do all the strain development until we're ready, and then we give the strain to the people that do the process development. It should start earlier on, because you bump into issues once you start doing process development, and then you need to go back to the strain engineering phase. And when you start doing that earlier on, you will win time.
And I say one question then that's specifically to your case study, and the person is asking, if the wild type productivity was already high, why were you modifying the strain in the first place?
Good question. So that's actually related to the problem with the variability that the market is looking for. So the wild type strain produces a mixture of lactonic and acidic sophroליpids, which are already applied. I didn't have time to explain, but for example, Ecover already applies them in their ecological cleaning solutions. But then there are thousands of surfactants on the market, which are fossil-based or palm-based. And all the companies applying those are actually looking for alternatives. But each sector or each application requires different functionality, and different functionality translates into different molecular structures. So the market is really looking for more variability. So that's what we try to do. We try to engineer the strain, the native strain, to make completely new structures, which are of interest to the market. But obviously then, typically the productivity drops. And then you have to start looking why it's dropping and what you can do to make it higher and to make it work and to make it affordable.
Great.
There's a few more questions, if I may, John. Is that all right? Or do you have a specific?
No. Let's ask these questions. If we ask the ones that are there, maybe one day we might move on, and if people have more questions later, we can come back to them for Sophie.
So Sophie, there's another question saying that besides the reasons already given that we should consider when designing for scaling up, are there other reasons why innovative microorganisms are not scalable? So are there other reasons why innovative microorganisms are not scalable?
Specific types would not be scalable. If for example, what I like an example that I encountered is, for example, if your strain is producing endotoxins, which is typical for bacterial organisms, for small molecules, that's no issue. You have ultrafiltration methods and other methods to remove the endotoxin, to separate the big polymer, which is the endotoxin, from your small biochemical. But if you're trying to produce polymers, that might become difficult. So we run into that in another biosurfactant project where the endotoxin has quite similar properties to the surfactant, which was a polymer. And then, yeah, it would have been better if the production strain was not a bacteria, or at least not a gram-negative bacteria producing this endotoxin. But that's something we also found out quite late in the project. And yeah, then, okay, you have to work with it and try to overcome it. Typically also what we've seen in the field, for example, there was a really interesting microbial biosurfactant called rhamnolipids, which were produced by a native strain. And actually, the native strain was a Pseudomonas aeruginosa, which produced quite high levels at high productivities. But you know, it's an opportunistic pathogen, and even though some people started producing industrial scale, it never really kicked off. And so now Evonik actually, they have developed a genetically modified strain that produces the rhamnolipids, which now enables them to build a multi-thousand scale facility to start producing these compounds, because there was an interest in them for a long time, but the fact that it was an opportunistic pathogen just didn't allow the industrial production because of too high risks. And there are others, but I can go on.
Thank you very much. There's one more question, but we're going to come back to that. And due to time, I think we should go on to the second speaker. Thank you very much, Sophie. Thank you very much for your very nice presentation.
Thanks a lot.
So if you could stop sharing your slides, Sophie, and maybe Olga can start sharing her slides.
Sure.
Yeah. And while they're doing this, I can introduce you to Olga. So Olga is from a partner from the TOPCAPI project, and she is the scientific director from Medina. And Olga, are you okay?
Yes, I think I'm here.
Yeah. Yes, I can see your slides. And if you can make it into a full screen.
Yeah, let's try if it works.
And she's going to be talking to us a little bit about the what Medina is doing and the analysis aspects.
It's already been in the presentation mode? No.
No, it's not. It's still in the, you can see too.
Let's see. And now?
Yes, it's
okay,
a presentation.
Perfect.
Thank you very much.
Thank you so much, Enrico. Pleasure to be here today. Yes, in fact, I will follow up on the different aspect of what we are doing in the TOPCAPI project, and we'll use different examples just to show how we can apply the compound analytics at the validation stage at different levels. So just to remind you what we are doing in the project, there is 8 different partners from 5 different countries. We are organized with a special focus in trying to engineer antibiotic high production chassis in Streptomyces. So this is leveraging, as you know, host engineering and microbial cell factory engineering tools to integrate all this system biology driven strain design and SynBio, as well as the industry level improved performance to establish actinomyces platforms. But also we are very much interested in pathway engineering, just to optimize the production, just on the basis of all the modeling that is being performed in the project, and at the end, just to obtain improved antimicrobials. So at the end, all the process, it means as Rico presented in the other sessions, is an iterative process where we are engaged into a system biology analysis and modeling that is also feeding the way strains are engineered to manage to get data that are also helping us to generate a lot of information that at the end are fed into the models. So as you see, the partners are engaged in different parts of this process. And today I will use one of the compounds that have been targeted in the project, and that is in fact the application of these tools of this compound analytics to optimize the production of this cytopeptide G2270, that in fact is just a precursor of 2 molecules that are now in the clinic, that are being in development with different applications. The point is that the compound is produced by Planobispora rosea, which is not tractable. with conventional methods. So any improvement of the production, that is one of the major targets in this case, cannot be performed directly on the white-up strain. So the first step is just to move all the biosynthetic cluster into a heterologous host and to manage to get expression and achieve a higher production of this compound and in higher titers. So at the end, when we are talking about compound analytics, just with a objective to test and validate the processes, we want to understand the physiology of the white-up strain, but also the recombinant clones, and to translate all the knowledge that we can obtain from the data that we are obtaining by profiling the metabolites, both within the cell, but also what is being secreted into the broth. We will also want to monitor and quantify the production of the targeted compound and all the congeners that are being produced with the different rounds of improvement. And at the end, having the tools to elucidate any new molecule and then define the structures of these new compounds. So at the end, just to perform all these processes, we rely on the metabolomics, the metabolomic platform that we had across the consortia and the different of the members, using low-res mass spectrometry. This can be coupled to UV analysis that is also giving us just in preliminary approaches a lot of information, but of course all the accurate analysis needs to be performed using high resolution mass spectrometry. And for this we can approach the use of different type of equipments. This can be just as an example the Q-Exactive, as in this case, or we have been using QTOF mass spectrometry. So this is also enriching because of course the different type of analysis can be performed, and this is helping us as well to validate the process that we are performing. On top of that, of course, we need to rely on NMR structural elucidation, all the spectra, to really get into the elucidation of the structures of the new compounds that we have in the process. So together with this, well, what we need to approach is the metabolomic approaches can be done in two different ways. I mean, when we are going to be studying the production of a non-compound, we will be focusing on a targeted metabolomics. In this way, we will try to track all the molecular ion, the fragments, the retention time, all the building blocks that will be generated during the process to really quantify the production. So at the end, we will be just focusing our work on trying to infer the production of the targeted molecules, any related components as well, and quantify along the time during the fermentation process. But at the same time, we want to learn from the process, from how the strains are behaving, what is their metabolism, the physiology, how this has been just reacting to the conditions we are exposing the strains during the cultivation. And in this case, we are using the untargeted metabolomics, where we don't know in general most of the components that we are going to be obtaining from the profile. So we will get not on top of the retention time from the separation, accurate masses and molecular formula, from where we will go into the databases and searching for matches and trying to infer the identification of these components. So these are the two ways that we have been using during the project to answer many questions according to the needs and the steps where we are in this iterative circle. So in this case, you have just an example of the results with the wild-type strains, Planobispora rosea, just targeted metabolomics, where we can track the production of the targeted compound, the antibiotic, but we also can track along the time during the fermentation, the production of the different congeners that can also show us how the strain is behaving and how all these small components in the production of these families of molecules is taking place along the time. So in this table, you see all the different chemical molecular formula for all these components in the broth. But together with that, we can also just question what is being produced at the same time and how the strain is behaving, to learn from the physiology and how we are feeding in the system with the different nutrients components, and at the same time just to understand which are the fluxomics within the cell with time. So this is normally being done using these untargeted metabolomics, using the an endo and exometabolome analysis with a cold methanol extraction in general. And then in this case, we will be annotating, just as was explained before in the extracts, the molecular formula, the retention time, and trying to infer from there the presence and absence and the trend with time. So what we see on the graph is just the evolution of the profiles with time and the appearance of the compound in the position together with reference to the standard. So this is the type of information that can be obtained from these untargeted metabolomics. And at the same time, when we go deeper into the analysis, we can group the metabolites according to the trend with a fermentation time. So in this case, you can see that we've managed to group that into 8 different groups with different trends, both in the extra and intracellular components, where you see that groups A and B on the left are really just components which are increasing and they are accumulating during the growth of the strains. The next groups from C to F, they are just corresponding to molecules that are being depleted at a certain point of the process. So this is also helping us just to understand what is the behavior of these components within the cell and also what is being secreted into the medium. One thing that was really attracting our attention is the behavior of the amino acids. We are just corresponding to the group G. And in this case, we saw the same pattern, just a decrease in the titers of the different amino acids, except in one of them that in this case, this was corresponding to glutamate that was increased in the case of the intracellular component. And this behavior was not seen in the extracellular amino acids. All these data were also correlated using other type of omic analysis in the cells. And at the same time, we will see the trend of the congeners from the antibiotic that was targeted, and is helping us just to understand how the wild type strain is behaving and correlate this analytical data with other omics data that are being performed and obtained during the process, and feeding the models and having a full picture of the wild type strain. Together with that, of course, we went of the same type of approaches have also been applied with recombinant strains. In this case, with Streptomyces silicolor, where the cluster was cloned, and one of the first analyses to show that in fact the production of the heterologous strain was very low compared to the wild type strains, and with higher levels of the components in the endo extras compared to the exo. So there was a problem with the secretion of the component. We also saw a lot of congeners that were related with cyclic and linear that were not explained in how they were produced a long time from the targeted metabolomics of all the related molecules associated to the major antibiotic. Compared to that, we also perform an untargeted approach just to see what else was being produced, and we compare both the Streptomyces silicolor devoid of any cluster and the heterologous host that was just producing the small amounts of the antibiotic, and to understand which were the trends for the other type of molecules that were also being produced by these strains. And as you can see, the exometabolites were increased in certain metabolites in the presence of the cluster. When on the other side, when we are just observing the endometabolome, there was a depletion when this cluster was introduced in the Streptomyces host. So this information was all collected and also used to be entered into the system to really understand which could be the dynamics of the host, and then which could be the bottlenecks preventing as well, and how could be improved for the production of the antibiotic. So not only we use Streptomyces silicolor, but also there was the use another host which was much more related taxonomically to the original producer, which is Nonomuraea. And in this case, one of the examples that I'm showing today is just how we were applying all the analytics to understand which were the nature of the compounds that were produced by these cosmids that were devoid of some part of the cluster. And in this deletion in this fragment of the biosynthetic gene was determining the loss of the capacity to form the pyridine and oxazoline rings that were inherent for the molecule. And they are shown in the graph. So when these cosmids were analyzed from a metabolomic approach, we identified that in fact there were 5 different components that were produced by these cosmids that we wanted to go more in depth to understand how the lack of these enzymes were just responsible for the production and what could be the potential structure for all these analogs that were produced by this defective pathway. So as you can see, these they were produced in different amounts, and we went into detail just to extract each of them from the spectra, and just to infer from not only from the exact mass but also MZ from the molecular ion, but at the same time the chemical formula. And you can see that in fact all the components that were detected, they have several specific features. They were not circular again. They had been losing the rigidity of this tetracyclic score of the pyridine, and they were resulting in linear compounds. They have been losing the C-terminal amide moiety. At the same time, there was an altered decoration that involved the methylation and the hydroxylation at different positions. So the deletion of these 2 genes were really affecting, and there were these shunt compounds that were produced from the biosynthetic gene cluster. And then this analysis, just to show you how we are using the analytics in this case, I will go more in depth in the peak number 4, which was the major peak that was obtained in this case, and how we use the analytics just to interpret the fragments that were obtained from the fragmentation of the peak. We had the molecular ion that was on the left, which was really maximized here at the bottom. And at the end, that we started to infer which could be the fragments from this spectra that could give us some hints about the composition of these molecules. So in the final graph, you can see how we can be applying all the analytics and all the information that mass spectrometry can provide just to help us to interpret from the spectra, which could be the fragments that we are seeing, and reconstructing the structure that we have that being produced by the strain. So in summary, as you have seen, there's been different type of applications of the mass spectrometry and the analytics in this case, and that has been obtained and has been just used in the different stages of the project, just to confirm, validate, and test that in fact the molecules that are being predicted and that have been targeted through the engineering of the strains are efficiently being produced, and also to monitor at the stage of the fermentation and the scale up that at the end you are producing the compounds in the titers that you are expecting. So these are tools today that need to be there at any point during the development, managing to get just a potential product close to the development stage and to move into a large scale production. So in summary, this is the major part of the analytics, and today this is something that is crucial to be used in this type of project, and also just to have all this information being fed into the models to help to predict and modulate the potential approaches that can be developed in this case. So finally, I would like to thank all the members of the consortium, and especially those that were involved in all the analytic studies on this study, which is not only from Medina, Mercedes, but also Camila, Oksana, that have been very much related involved into the generation of all the data that were presented today, but also all the partners that contributed to development all the tools and the materials that were tested during this study. And I will stay here, and I will take any questions. Really happy that you may have. Thank you.
Thank you very much, Olga, for a nice presentation and overview on the G2270 production and validation. And as Sophie had said at the very beginning, how important it is, analytics is so important that we can actually see what we're producing, isn't it? So that was great. Let me just check. So please, if you have any questions, please put them into the question and answer box. There's a question box so that I can read them out for you. And before we see any questions here, maybe I can ask some questions, Olga. I know that Medina is specialized in looking at all sorts of natural products, and I know you have lots of projects going on at the same time and looking at lots of different analytical techniques. What do you think is the most futuristic analytical techniques? I know we all use LCMS, GCMS, NMR, maybe not as much these days, but do you see any of these technologies that is quite, standing out, coming out in the future, and that you think would be like a good workhorse that everyone might start using?
Well, in our case, of course, just not only just mass spectrometry, but also NMR. I mean, we are using both of them in parallel, and many times with very little information, and even not even with the compounds purify, and then we can just integrate all these approaches very efficiently together with the use of the database that we have in house. So this is really, really helping us a lot in managing to get very quickly to know if we are dealing with a non or a non-compound. So this is probably most of our interest in the case of the type of work that we are doing. Of course, there is a lot of exploratory work at this moment. We are doing a lot of prospective metabolomic studies, trying to infer from the knowledge without having to test, and exploring as well how to manage artificial intelligence in getting all these deep learning processes and integrating all the data that we have here, just to help us, just to minimize the benchtop testing or just wet lab approaches, and having some predictions beforehand, just leveraging all the data that we have, but also just coupling this to the information that we may have in terms of activity, and trying to correlate that, because at the end we are also working with complex mixtures, and this is one level of complexity that we have in the analysis. So this is something that we are very, very much interested in. And of course, we are integrated in anything that the community is implementing in terms of using these tools to connect and networking compounds, just to manage to get very quickly rid of things that we are not interested to work with, and focusing on the most interesting molecules. Yeah.
Do you think industry will be also interested in these kind of databases and using artificial intelligence to actually try and predict? Because actually, if you think about it, it's obvious, isn't it, that we should go to this next stage, because you have so much data. And if you think about all the community putting together all this data, that would also become quite a big asset, I guess. Do you think industry would also be very interested in something like this?
I think so. I mean, when we are talking about industry, it's not only just traditionally what we have seen in terms of the pharma companies that were just doing natural products in the past. They are no longer working so much as such. Probably they will come back with a different perspective and different interest in the future. But also there are many other sectors of industry that have a lot of interest in trying to really profile and getting deeper into that. And there are so many applications that they were just presented as well by Sophie, that at the end you have microbial small molecules. At the end you have a chemical space that you want to depict, and with many different type of applications. So I think this is a very, very unique area to go that will be expanded for sure, and databases will be there always just to help.
Yeah. Okay, thank you. John, did you have?
Yeah, I was going to, Olga, enjoy that. And I mean, you showed in a couple of the figures that was a potential, I think, especially for the strain with the heterologous engineering for other metabolites to be produced also. And you talked about generating the data to feed the models. And I was thinking back to some of the things that Sophie was saying. And I'm just wondering, maybe in the TOPCAPI project as a whole, I mean, to what extent did did you try, using some of the methods that you described there, some of the kind of what I'd call the high-powered analytics, also at the production stage or at the stage of process development? And were you looking at that kind of integration that Sophie was talking about, or was your approach maybe a little bit different?
Well, at this stage, in fact, most of the times we have very little information about the physiology of the strains, and I think it's the first layer in trying to build the most basic data to fill the system. So these iterative systems are helping just to infer which are the trends within the cells in the different contexts. Even switching from the wild type strain, is a completely unrelated genus, to the Streptomyces, is a completely different behavior. Even the same biosynthetic cluster is not behaving the same. And ourselves at Medina, we are experiencing the similar cases, and we know so little when we move out from the model organisms about how to manage and how to refactor, how to influence, which are the bottlenecks. And the major focus was to try to understand what is ongoing because if you don't have the information, I mean, you don't have the basis just to build on that. So that's the major limitations when you try to optimize and to scale up. Many times is a lot of serendipity into what you're doing, because of course you try to guide the process, but you are in a context that you don't control at all. I mean, not at least completely. So I don't know if Erika's also, because we can also have this discussion later, but I think we are facing so many cases where you fail, and that's the problem, because in fact you know very little from the wild type context where this compound was produced. So that's,
I think it's really difficult. What Sophie said sounds easy, but in fact it's just so difficult to do.
Yeah.
And with several projects that I organized, we always try and say, okay, we have to think about large scale production to be able to teach the lab-based wet design. But so far, it's not 100%. We are not there yet, like what Sophie's saying. We should be, but we're not there yet, I think. But it'll change. I think we will come to a time where we'll change.
Yeah, I totally agree. Yes, we are just going at as high as 15, 30 liters ourselves. And that's a big challenge many times. And also having to deal with the filamenting microbial strains, where you have a lot of also limitations that probably are added in this case to the problem of producing the targeted compound. Yeah.
And Sophie put in a comment in the chat there, I think, which is very valid. I mean, even being aware that it's an issue is already a start. And I think that's certainly true. Even, I was going to say, even if you're doing nothing about it, but even if you can't address it right on, just be aware of it there in the background so that it can be addressed when possible is better than being oblivious. Well, I sometimes think ignorance is bliss, but maybe not when you're trying to develop a bioprocess.
Yeah.
Yes, right. Thank you so much, Olga.
Thank you. Pleasure.
With the time, I think we should move on. John, is that okay? Maybe we can ask more questions later on.
Sure. Yeah. Let's move on.
Yeah. We stopped sharing. I'm sorry.
Thank you very much, Olga. Thank you very much. And
thank you.
So, if Paolo could share his presentation, Paolo.
Yes, I can.
Doing that, I can introduce Paolo. Paolo is a partner from the CHASSY consortium, and he is in Clarence Business Line Biofuels and Derivatives, which develops and commercializes cellulosic biofuels and bio-based renewables for the Swiss specialty chemicals company Clariant. So without further ado, Paolo, if you could please start your presentation.
Thanks, Erika. Just tell me if the slide is showed in the correct way.
Yes.
Excellent. So thanks to the organizers, thanks also to the people attending the webinar, and for the time that you are dedicating to the session. I wanted to start by saying a warm welcome from my side, but I'm based in Munich, Germany. We're almost in the middle of April, and until yesterday we had the last 3 days of snow. So maybe I shouldn't start this way, although for the presentation that I'm having today, I will be focusing on a technology solution that has a strong impact on the environment. So there's a clear link. So I will be focusing on our technology, which is called Sunliquid. It's an advanced cellulosic ethanol technology. I will also guide you through a refresh on the legislation, so the reasons why there's a support from the legislation side into these technologies, and then a deeper dive on the technical details and also commercial readiness. I work for Clariant. Clariant is one of the world's leading specialty chemical companies. We are Swiss-based, 3.8 billion Swiss franc sales in 2020, more than 13,000 employees worldwide, and we have more than 80 production sites. Within Clariant, innovation, R&D, and sustainability do play a very important role. And these are all aspects that are very much linked to the technology solution that I'll be presenting you today. You've been hearing the previous presenters talking about R&D aspects, talking about upscaling technologies, also value of death. We went through all this. We started the concept on this technology back in 2006. In 2009, we built up a pilot facility in Munich. In 2012, we started up our pre-commercial site plant in Germany. I will talk more about that later on. And as we speak, we're constructing our commercial site plant in Romania. I want to show you, and I want to start this presentation by showing a short video of this of the construction of the site. But before doing that, let me tell you that this is a commercial plant that will be producing 50,000 tons of cellulosic ethanol using agricultural residues as feedstock. So this is why we call it an advanced cellulosic ethanol technology. And I'm proud to say that safety has been one of the key elements throughout this time of the construction of the plant. We have, as we speak, 750 people on the site working on a daily basis and engaged in the completion of the facility. And safety has been a very key element, especially throughout these unprecedented COVID times. We reached 1 million hours of work without any lost time accidents. So let me start the video now, and you should also hear music, and I will talk more about that later. So please keep in mind that, as I told you before, we have been developing a concept back in 2006, and we wanted to use agricultural residues mainly as feedstock to be converted into sugar, fermented into ethanol to produce a commodity. And now we went through all this journey, and we're actually deploying this technology on a commercial scale. This is a facility. This is a commercial site plant. This is fully financed by Clariant. It will be operated by Clariant, and it will serve as also a training facility with our customers. I want to quickly walk you through the actual process. I think it was important to do it on a picture of this site. So this is a site in southwest Romania, in the town of Podari. It's roughly 10 hectares. and it will be completed within this year. So we will start production with this year. And as you can see, all main equipment and also the main buildings have been erected. What we will do is we will source mainly wheat straw and barley straw from the neighboring region, from around the plant. We will have a small straw storage area. The straw will then undergo a first mechanical thermal pretreatment, which is basically taking the ropes out of the bales and milling the straw in smaller particles, going through a steam exploder. This will enable the first step, which is the hydrolysis step, where enzymes, these are process integrated enzyme being produced in the at the site. These enzymes will disrupt the fibers, will extract the sugars, mainly C5 and C6 sugars. And we have this first step in which we have a solid liquid phase, biomass and liquid phase that has to be filtered. So the biomass is filtered out from the liquid phase, which is mainly lignin. Lignin can go in this CHP power plant, so combined heat and power plant, that will provide the energy, so steam electricity requirements to the facility. Once we have these filtered C5 and C6 sugars, these sugars are then fermented into ethanol, into this next step, the ethanol fermenters. This is done in a one-pot reaction, so both C5 and C6 sugars are fermented into ethanol. We have then a final stage, which is the distillation area. This is undergoing a beer column rectification unit and a dehydration unit to reach the ethanol purity of 99.8%, which is a fuel grade to be used, for instance, in transportation. Ethanol is stored here, and this is, I would say, in a nutshell, the main process steps. It is important to highlight the fact that we have a completely chemical-free pretreatment, the fact that both the enzymes and the microorganisms, so the yeasts, are fully process integrated in the facility. This has important aspects not only on yield improvement because they're feedstock specific, but also on the cost aspects. So some additional data on the plant. This will be producing 50,000 tons of cellulosic ethanol per annum. It will require roughly 250,000 tons of agricultural residues being sourced around the area. And here we're talking mainly wheat straw and barley straw. The site is a 10-hectare site, including the power plant for the energy requirements. The overall greenhouse gas saving is 95% compared to gasoline, so highly sustainable. This number would go even higher to 120% if we capture CO2. CO2 is another side stream out of the fermentation process, and that would go then into a carbon neutral end product. We have another side product, which is vinasse, which is the distillation side product, which is rich in minerals, NPK. So these minerals can either go back in the field as a biofertilizer or can be used as feedstock for biogas use or for additional energy being produced. The final product is ethanol for transportation use. It's been off-taken, so oil major will be bringing this product to the market. And as I said, this facility will be utilized as not only a commercial production site, flagship, but also as a training facility for our customers. My task, or one of my tasks, is to bring this technology to the market. As of today, we already sold 5 licenses, 3 in Europe, and I'm starting here with the first one being sold to a Slovakian player. It's a big biofuel player. So we will have an integrated second-generation ethanol plant to an existing first-generation ethanol facility. This will also be a 50,000 tons ethanol plant utilizing locally sourced wheat straw. The second license has been sold in Poland to a refinery, to Orlen Południe. So this will be mainly an energy integration, second generation plant to the existing refinery. It will be a smaller size, a 25,000 tons ethanol per annum, using mainly wheat straw. The third license in Europe has been sold in Bulgaria to a big agricultural company. The project is called ETABIO, and this will also be a 50,000 tons ethanol facility utilizing wheat straw sourced locally. And then finally, we have 2 licenses sold in China. One has been sold to the joint venture of Guozhen Chemtex for a 50 KTA, upscalable to double the size, using corn stover and wheat straw. And the last license being sold during these COVID times, all virtually, without physical meetings, has been sold to Sinodan. This will also be a 25 KTA using corn stover. This is important to say that there's an interest from different players in the value chain. So I started talking about the agricultural side to the first generation players or biofuel players, to the mandated parties, which are the mineral players. How does legislation play a role in this, in the deployment of these, in the demand of these technologies? We're focusing in Europe. There's a clear legislation. It's called the Renewable Energy Directive, that requires mandated parties or mineral players that put into consumption mineral fuel to blend certain percentages of fuels, not only advanced biofuels, but also the so-called first generation biofuels, throughout the pathway, starting as we speak until 2030. And if you see here the blue area, this is the requirement to blend in advanced biofuels. It's actually a minimum target starting 2022 with at least 0.2% of all the mineral fuels put into consumption. This percentage is going to 1% in 2025, up to 3.5% by 2030. What does this mean? This means that these players are mandated, so each member state needs to implement this legislation at member states. It would be done within latest June of this year. They have different type of feedstocks that can be utilized, which are part of the so-called Annex 9A. Here I'm listing some of these feedstocks, which are all feedstocks that we have been treating and we can convert with our Sunliquid technology. There's different baskets and different requirements to comply with the Renewable Energy Directive. Annex 9 feedstock, which is not considered advanced. These are mainly used cooking oil and animal fats, and then the conventional biofuels, so mainly the so-called corn or seed from first generation. In terms of volumes, we're talking 5 to 10 million tons of product in the market, so substantial numbers. Why did I want to focus on the fact that we have an integrated enzyme production? Because of different aspects. How does this differentiate from off-site or on-site enzyme production? Well, looking first on off-site, here basically you have a production plant, ethanol production plant that needs to be sourced from third-party providers on enzymes, so big volumes, constraints on when it comes to logistics, cost, purification, and concentration is required. An on-site production is you have close to it, maybe off the fence, but still you need, for instance, glucose or other more expensive nutrients to produce this. While the integrated uses, like Clarent case, uses a very cheap biomass, the same agricultural residues as a carbon source and small other nutrients, and it's all integrated in the system. The same goes for the yeast that are being produced. So this has important aspects. Not only that, as I said, we use as a carbon source the same feedstock, so this has impacts on the cost, We have feedstock-specific enzymes and microorganisms. No logistics costs are being utilized. No purification of concentration is required. And this has then very important cost elements effects, where our technology then enables to reduce substantially cost on the integrated enzymes. Similar considerations goes for the yeast strain. Here on the left, if we were to compare our Sunliquid yeast strain to competitors for the production of the same amount of ethanol, then you would see that thanks to the fact that we do recycle yeast, we're having a 70% lower yeast dosing versus competitors. And on the right, when it comes to production cost, so Euroliter ethanol, the same also is true. We have a substantial decrease in yeast ethanol production cost. And this is just to mention two elements that have important aspects on the overall production cost. I talked to you on the fact that we scaled up the concept from lab to pilot to pre-commercial scale. This is a picture of the pre-commercial scale plant in Germany. We have been validating, fully validated technology for more than 8 years using different type of feedstocks. We scaled down from an engineering process to 1,000 tons of ethanol per annum facility. We tested different straws, energy crops, rice husk, but also bagasse and sugarcane tops and leaves. And this serves as a training facility, but it also serves when we invite our prospects to bring their own feedstock, and the results obtained are part of the license details that we actually bring to the market. Different type of feedstocks being converted. Here you can see some of these. Wheat straw, corn stover, woody residues, rice straw, empty fruit bunches. These have all been tested in Germany, and for these, we're ready to deploy the technology at commercial scale, including process guarantees. And we have additional more than 30 residues being tested at lab and pilot scale. Last slide, let me conclude on the fact that how can we support prospects throughout the value chain? We support them from the early beginning on the assessment, when they have a product assessment, into partners and marketing support. Throughout the development, depending on the type of feedstock that they use, we can also do testing in our facilities. And the natural next step is then take a decision to go into a contractual negotiation, where we'll provide the basic engineering, the licensing for the utilization of this technology, and the starting culture. So we provide very small volumes of starting cultures that enable them always to have fresh microorganisms to produce yeast and enzymes throughout the journey until the project and operation of the facility. So we always keep a very close contact to our prospects. Let me stop here. I want to thank you for the attention. I'm ready to answer any questions should you have.
Thank you very much, Paolo. Thank you. I think Sophie had a question. Sophie, I know you wrote something into the chat. Perhaps you would like to unmute yourself and ask the question, if you can.
Yes. Okay. I thank you for the nice talk, the nice information. It's nice to hear that this is really finally happening. I was wondering, you mentioned costs, but I don't know if we're comparing to second generation competitors or first generation. And yeah, typically using these side streams, it makes the final product more expensive. And my question was, how was that for you? And will you be able, if it's more expensive, to put it on the market more expensive than a first generation ethanol, because it's derived from lignocellulose?
Thanks for the question. Yes, I was comparing costs to second generation alternatives. Yes, you're right. These are new advanced technologies. It also has additional steps if we compare this to first generation. For instance, the treatment of the agricultural residues, the steam exposure, part of the hydrolysis step. These are new steps that would need to be added. And yes, the production cost can be more expensive versus a first generation ethanol production. But it's also true that the feedstock being utilized is a lot less expensive from corn, for instance, from the crops. We have regions, I'm thinking of China, for instance, where the second generation production cost is actually less expensive than first generation ethanol. So it very much depends on these factors on one side. On the other side, it's also true that the final product, for instance, I can mention the example of Europe, is being paid at a premium versus the first generation products. So I hope that this answers your question.
Yes, thank you.
Thanks. John, did you have?
Gosh, I could ask a lot of questions here, but I was just thinking at the end, Paolo, and I enjoyed the talk, and I thought your discussion of the integrated nature of the facility where you're producing the enzymes on site, and you know, not having to bring them in. But then you talked about trying different feedstocks, and you know, customers could come and try whatever feedstock they might have available. And I suppose my first question just on this is, you know, to what extent is the system modifiable, or is it a, I mean, is the only modification how you might, let's say, pre-treat the feedstock, or actually is there scope or indeed need maybe to modify, I don't know, the range of enzymes produced on, or the yeast strain, or other aspects of the process?
Yeah. We always refer to hardware and software. The software, it's something that we provide, and it's what I said, it's the starting cultures. Starting cultures are feedstock specific. This is something that we have been developing in Clariant. We have a high throughput screening lab. So we always provide feedstock specific starting cultures to our clients. So they can change the type of feedstock as an inlet, and they always use the adopted starting culture. It is true that this is the software. It is true that from a hardware point of view, it depends what feedstock are we talking about. If I go back into this slide here, you see, for instance, woody residues do differ quite a lot versus wheat straw bales, or even bagasse, which is usually a loose product, differs from a straw bale. So in that case, yes, the mechanical pretreatment would need to be changed. But if we, for instance, if we talk about the Romanian facility, I mentioned at least two feedstocks. We have wheat straw and barley straw being processed. The technology and the setup, so the mechanical setup will not change. Yes, the software, so the enzymes and microorganisms will change and will be adapted to the feedstock of choice. So the technology can process different type of feedstocks. If you're talking about two different completely type of biomasses, then yes, the pretreatment, the mechanical pretreatment can change, and some of the parameters have to be adapted. But I think that one of the important aspects is that you need to have a technology that is feedstock flexible because of price fluctuations, because of volume availability fluctuations of the feedstock. So you have to have a flexibility.
And thanks, Paolo. And I see somebody's asked what kind of I guess a follow-up question to that, because it's talking about the yeast strain, and it asks a few questions. It asks whether the yeast strain is also used for enzyme production. And then it asks, is the strain you're using a modified C6 yeast, so a hexose fermenter for C5 consumption, or is it a native C5, C6 consumer?
Yeah, so we actually started with non-GMO strains, but then we soon realized that with GMO strains, better yields can be obtained. As I said in my slides, the enzyme is able to extract both C5 and C6 sugars, and both C5 and C6 sugars are then converted in one-pot reaction in the fermentation step by proprietary yeasts.
Mm-hmm. And actually, just on that, because I know in some of the first generation plants, the yeast are kind of I don't know, used afterwards for other products, animal feed or other things. What can you do with a genetically modified yeast at the end of the process?
Yeah, so first of all, we do recycle them. As I said, that has impacts on cost reduction, but then they're completely deactivated. We don't have any issues when it comes to side products being brought back in the process chain, production chain, either via, for instance, the vinasse, as I said, going back in the field as a biofertilizer or as an energy source or in biogas production. So that's not a concern.
Mm-hmm.
There's another question, perhaps, if I can jump in here.
Sure.
Denise is saying, thank you for bringing this to market, especially without the need for chemical pre-treatment. She's agreeing with Sophie. And she's also saying, what do you use to do your calculations for the CO2 capture?
Mm-hmm. So good question. We have been following all the parameters that are part of the Renewable Energy Directive. There's a lot of annexes to that, so we're following that. When I mentioned the 95% greenhouse gas saving, or when I mentioned 120% greenhouse gas saving, that's what I'm referring to. On the 120%, that's when we capture and eventually utilize the CO2 for alternative uses. That's when we go into carbon neutrality or even negative carbon emission.
Oh, great, good. There's another question. Is there a prospect for going to a complete consolidated bioprocessing approach with your setup?
I'm not really sure I understand the question.
I think that the person is probably asking whether you could have the yeast that's carrying out the fermentations, could that also produce the enzymes?
So a simultaneous
yeah.
Yes, that's also something that we are assessing. Yes, it's something that can be done, although the approach that we took, and it was actually the title of the presentation, the approach that we took is to have these separate, because then we can commercialize what we call the sugar platform. So once we convert the residues into sugars, those C5 and C6 sugars can be converted or fermented into other bioproducts, not necessarily fermented into ethanol. And that's actually what we are commercializing also as a platform. Once we do the SSF, then that wouldn't be the case.
Yeah.
There's one more question from Alina. What are specific legal requirements or limitations for using GMO strain for ethanol production? Any legal requirements or limitations?
No, we don't see any legal requirements or limitations. It is sometimes very much linked to a regional requirements. Certainly, this is no limitation in the countries where we have been selling our licenses. So Europe, China, for instance.
I see. Okay, maybe we can stop here and go into general discussion. Thank you very much, Paolo, for your very nice talk. If you could stop sharing your presentation. Thank you. maybe we can get into a more general discussion. And before we do that, because there was one question for Sophie, if I remember right, which we could not ask because there was.
Yeah,
Which one was that one? Oh yeah, here it is. When you collaborate with universities to improve strains, how long do they usually? How long do they take? Huh? How long do they take usually to come back with results? And is that data often published? To Sophie, this was a question, Sophie. So when you collaborate with university to improve strains, how long do they take, and to come back with results? And does this data get often published?
It really depends on what needs to be done. Typically, when we work with universities, that's in these consortium-based projects, so Horizon 2020, BBI. So typically it's done within the framework of the project and the time that was set forward to do a certain task. But obviously, you often run into unexpected results, which then require unexpected work to be done. And then it's really up to the consortium to decide what should be focused on and how we can tackle certain issues. Can take a year, and sometimes typically the projects that I coordinate we do it in an integrated approach, as I explained. So throughout the entire project we are doing genetic optimization and engineering, so from the start until the very end, and then the next project starts.
Right. And do you publish these data usually?
Yes, depends. So we often, of course, also patent strains and technologies. But then after a patent is filed, it's typically also published, yes. But
Okay.
not everything is always published, but typically most of the information is made available, yes.
Okay, great. Thank you very much. Okay, so maybe we can now have a bit more of a general discussion. John, do you want to start, or shall I start?
Well, I've got a question that actually
Okay.
for both, maybe especially for Sophie and Paolo, but anybody else who feels they have a comment, I'd welcome as well. But and I guess it's more on the commercialization or the marketing side. And my question is whether there is a premium for sustainability. In you know, I had a discussion this morning with actually about with a commercialization specialist about an idea I had, and I was arguing because we were going to be producing a particular product from a waste stream, that this kind of added value to the whole thing, and the person was saying, well, only if it's still cheaper than making it from a more conventional feedstock. And so I guess that's my question, because I think for Paolo and the bioethanol, where you can get higher price for advanced biofuels, there is. But maybe in a more general setting, I mean, is there, I'll call it a premium or a value to being sustainable in your production?
Mm-hmm. Yes, well, you answered part of the question. I think it's clear on the cellulosic ethanol for fuel use. I gave the example of Europe, where there's clear mandates. If there's a mandate, there's a penalty. And if there's a penalty, then there's at least a benchmark that creates a premium for these advanced biofuels, especially now that there's a very low production put into the market. So there's a supply-demand imbalance. From the other point of view, maybe ethanol or even sugars for other users, where maybe there's no legislation, or there's not yet a legislation for other biochemicals users. I think it's very much driven by a market pool. Yes, there are players that are strategically looking into these molecules because of the high CO2 footprint, so greenhouse gas saving again, because of the fact that they are not linked to the food value chain and using waste or residues. So yes, there is a lot of interest to go into this direction. This is what we see on the market currently on different products.
Thanks. And Sophie, what would you say?
Yeah, I completely agree. And I also think we often have this discussion internally as well. It's something for legislators. It's something, the thing that happened for biofuels, it could also happen indeed for biochemicals. And I think this could be really a game changer, because when legislation changes, that actually made biofuels happen, because of the incentives that were given, at least in Europe. I don't know the details about the rest of the world, but when legislation changes, everything changes. And I also agree with Paolo that it's now really through the sustainability aspect that companies want to advocate and sell to the customers. It's happening. And why is it happening? Because customers, consumers are really putting pressure now on big companies through the use of social media, which is something that didn't exist 6 to 10 years ago. And that really changed. And we really saw that 15 years ago, the companies that were interested in it, but it was too difficult, it's too expensive. And yeah, okay, maybe someday. But now that the consumer is really more aware and is having a forum through the internet, we see indeed that there's a dramatic shift. And I think a lot of companies realize that they have to act now. If not, they will be extinct in the future.
Okay, well that's
Very
good
interesting. Yeah. Olga, do you see any sustainability also importance in the natural products area? You're muted.
Well, it depends on these small molecules, the type of application that you may have. If this is a high value molecules that develop later with application in the clinic, it's a completely different market in this. But for other more just primary chemicals that can be also issued from microbial strains, at the end you will have an impact as well in the process. So this depends where the type of market you want to deserve, for sure.
Thanks. Thank you. John, do you want to go on?
Yeah, well, Paula, I have another question for you that's maybe it's related to this or not. I don't know, but I think you mentioned early on that something like 2006 was maybe when Clariant started looking at this, and you're getting into your big production plants now, and I guess probably vast amounts of money might be made in the future, but there's also a vast amount has presumably been invested. And for, one hears in the biopharma sector, very long lead times for, obviously not for COVID-19 vaccine, but typically for new products. I mean, for a company like Clariant, who's, I guess, coming from more, I'll say, from a chemical company background, perhaps, I mean, what was the thinking? I mean, was there a big strategic decision? And did somebody say, look, we realize that this is going to be kind of a long play and it's going to take 10 or 15 years to make money, but we want to do it? Or I suppose, what was the thinking in the company behind it all?
Yeah, I think it was two main elements. One is it was decided back then that they wanted to bring in a green footprint within their chemical business. That was one point. The second point is that they started back in the Sud Chemie area, and we were the same way we still are in Clariant, producing catalysts, and it was also an easy shift, I would say, to go from catalysts to biocatalysts. And that was really one of the reasons why we started this journey on the concept. How can we utilize this, the knowledge that we already have, and implement it and come up with a solution, and work with a huge abundant natural resources, which is agricultural residues, to convert that into sugar, from a sugar to a commodity. It's also true that the sugars can be then converted into biochemicals. So that's, again, you do see an angle to a specialty chemical company such as Clariant.
Mm-hmm.
If I may, I have a more general question to all three of you. It might be a little bit more vague kind of question, but everywhere I go, a lot of the countries, the funding bodies are talking about, you know, sustainability. But when you think about sustainability, it's to do with industrial biotechnology. Now, at one point, there was lots of things going on with biotech. Everything was going with gene modifications, so on and so forth, and everything was kind of going into this industrial biotech area. Nowadays, I have this feeling, I don't know if could be just me, that somehow industrial biotech has come kind of, not low tech, but kind of the fire's gone out a bit. Do you have the same feeling, or do you think that, you know, industrial biotech is still going strong? Maybe people don't use the word industrial biotechnology per se, but do you think that there is still a future out there for this kind of technology-based, bio-based technology? Especially, we always, you know, write in grants, petrochemicals, we're not going to be able to use petrochemicals anymore, but somehow we're still going on using petrochemicals because it's just so much cheaper. With climate change, you know, we're saying, oh, we can't do this anymore. We have to look after the climate, but we're still going on and using all these solvents and things. So I just wondered what you thought. Do we have a future? Do we have to do something to get it even more accelerating a bit more, or are we doing something wrong here? I just wondered what you thought.
We should start.
Yeah, you start, Sophie. Defend the sector.
Yeah, I don't agree with you that the fire died out. If you remember from my presentation, in 10 years we grew from nothing to more than 100 employees, and we are typically doing only biotech or working for biotech companies and universities. So yeah, I think it is really happening. And maybe to give you an answer on what are we doing wrong, I don't think the sector as such is specifically doing something wrong, but I think legislation can really have an impact. So I mentioned it before. If there are more incentives to go green, if companies are forced to go green, not only through the consumer but also through legislation or through incentives, that could really make the biggest difference. And I think this is something that is ongoing. For example, the REACH legislation that has been set up the past 10 to 15 years has forced companies to drop certain products in their portfolio because they had to start doing tests that they didn't have to do before. And that then opens up again some opportunities, because if a company drops a certain chemical from the portfolio, that opens up money, resources to develop new ones. Yeah, and then obviously the thing that we can do better is, yeah, it's still a young technology. Yeah, so if you would have looked at fossil or petrochemical industry 150 years ago, they also needed to go through a lot of learning curves and get the processes better, get them cheaper. That's what we now have to fight against something that is fully optimized, that is all the equipment is depreciated typically. So yeah, it's a full grown industry. Yeah, but as my boss often says, the really hardcore chemical industry, if they don't make a shift, they will be dinosaurs. They will be extinct in the future. So there's one thing that is for sure. Yeah, the fossil resources at a certain point they will run out. So and even we should not wait for that. As you mentioned, the sustainability, the environment. I think there is enough proof out there that we should not keep on using up the resources that we still have, but that we have to focus on the new technologies and start making it happen. Indeed. That's my point
great.
of view.
Yeah, thank you. Thanks. Yeah. Paolo and Olga, if you have...
I've not much to add to what Sophie already said. I also disagree that this the fire is basically gone. I actually do see something which is quite the opposite, not only on the oil majors that have to do this, but in a lot of other companies, the need to strategically look into this and really have the word sustainability as part of the DNA is something which is unprecedented and not seen before. I mean, you can Google the world and you see how many companies are actually focusing into this. Yes, it is true that the market, and now I'm talking more specifically on the cellulosic part, the market is also looking for this to be commercially viable and for this technology to be deployed at commercial scale, because there are companies that unfortunately failed. So we took a completely different journey. We did the homework in a different way, and we strongly believe that we're doing it the right way. And this is why we are going to bring this technology and this commercial scale plant within 2021 to production, and to show that this is viable.
Mm-hmm.
I think as well, Enrico. I mean, I'm burnt out. I don't know, is that the same as a fire being gone out? But I think, you know, if one looks at like the coronavirus story now, how science has really become in the public domain with that story. I mean, I think that's something that maybe they were missing, and that's why I can give rise to the kind of the perception you have, that people don't necessarily realize that sustainability is a science question. And even more than that, I would say it's a microbiology question, you know, because microbes really are at the heart of all these sustainable bioprocesses. And I think, you know, there's a communication gap there that somehow we, you know, we need to get our message out there. We need to get the success stories out there. So something like the, you know, the stories that Paolo presented there now in the Sunliquid, and the fact that this platform could produce loads of other things in the future are some of these really interesting surfactants that Sophie talked about, and things that are really successes. We need these to be almost public knowledge. You know, people need to know that science and microbiology is what's going to give rise to this sustainability. I mean, because I think at the moment it's a little bit of an abstract concept for the public.
Is, yeah.
You know, they know
it's like a name dropping. It's the same thing, name dropping in a website, isn't it? We're doing sustainability. What are you actually doing? What is sustainability actually, if you think about it? Yeah.
And so that's, of course, you know, speaking with my EU project hat on, you know, that's one of the things that you know, we're meant to do in EU projects. So if that information isn't out there, it's all of our fault as well, you know, because that's, you know, we have to try and communicate the messages and have to try and engage people, but it's not easy though.
Perhaps also it means that we should be lobbying into the EU for legislation, stronger legislation perhaps, and that might change a lot of things as well, perhaps. I don't know. I'm just saying, if legislation is the key.
Changing it. I think if you look to the CO2 story, there you see it quite clearly. Once they start charging per ton of CO2, then people start to wake up. So that is really key. I think that is one when people have to change, because change is difficult. Everybody says they want to change and they want innovation, but it's a lot easier to stay as you are doing it than to change, especially for big companies. It's extremely difficult.
And actually, can I ask a question for I mean, this applies to all three people now, maybe you could comment, but I mean, because of course there's the big chemical companies, and if they don't change, you know, they're risking their future. But how easy is it for small companies to get into the sector? Because, you know, you've got all these EU projects, you know, Sophie, you mentioned that you're involved in a good few, and obviously we've got CHASSY that I'm involved in, and TOPCAPI that Enrico is coordinating. And these projects are producing, you know, technologies that could have value and that are exploitable. So how, so I mean, if you're, let's say, a startup company or a small company, is this a difficult sector to get into, or what's the way to do that, or what's the best way to try and get this technology, I suppose, commercialized or just used?
Should I answer, or somebody else wants to maybe answer?
Go ahead. Go ahead.
Yeah, we are actually thinking about setting up a spin-off company to start commercializing the biosurfactant. So it's not easy at all, but it's not related to the sector. I think starting up something new is not easy, never. And I think we are privileged in a way because, yeah, it is, like you say, Eriko, it is a buzzword. The sustainability. There are a lot of funds out there to get going, but it's still extremely difficult because a regulation, for example, REACH, to put a new compound on the market, it's extremely expensive. You're looking into hundreds of thousands, millions of euros just for the registration of a new product. So also there might be some things in the future that can be changed when you want to put something on the market that has really high sustainability outcomes. So good LCA, for example, that there could be some incentives of some way, or that the registration can be cheaper in a way to actually promote this. But that's not easy either. It's yeah, it is complicated, but I think the means are out there to help new businesses. And as you say, there are a lot of new businesses starting from funded projects from universities. So I think we should keep on doing the good work, and as such, new things are happening and can go to the market. And then there can be collaboration between small companies and the bigger ones, because for the bigger ones it's sometimes difficult to make really a drastic change, but small companies are more agile, but they need bigger companies for market access, for the knowledge on the market. So that's how we try to do it, to really find nice collaborative models.
Yeah,
Olga,
I totally,
Paolo?
I totally agree with Sophie in the sense that at the end, this project, the small companies are much more dynamic and they can manage to get innovation much easier than the larger ones. That the larger ones are just looking at what is happening, and they will be helping to develop into that. But of course, what needs, and this has already been mentioned, I'm not saying anything new. I mean, you have on one side the question of the price, that just to be competitive enough, and that the market is driving everything. Investors and money is what they take the last decision. But at the same time, the end user, the consumer, also needs to get the right information and demanding and having this penetration of the new products in their day-to-day use of these products. So that's something that needs to work in both ways, at the beginning of the chain of value and at the end as well, being able to not only just to push ahead, but also to pull from the consumer and demanding, as it has been said. So that's something, and also to understand what the advantage and how this can be competing, because otherwise you get what you get into the market as user, and you don't demand that these products are being replacing those that are much easier being produced and cheaper for the large companies. So it's a trend, needs to be forced to some extent. And you have been mentioning as well the question of legislation, that at the end makes a big change in anything. So the big barriers for registration are huge for the small companies, and this is something that cannot approach in all the sectors.
Paolo, would you like to add something?
I certainly share the same thoughts. Maybe one specific example. Yes, entering the field with the construction of such an industrial facility for a small company, a very small company or a startup, can be a bit of a burden. However, we do see that there's a lot of smaller companies, for instance, that are interested in developing solutions for instance, on the side streams. So I mentioned lignin as a side product that currently is being used to be burned and provide the utility requirements to the plant. We do see a lot of companies that are actually developing solutions for higher value-added users of lignin. The same holds true for vinasse, for instance, this distillation side stream, or even for CO2. So there's different ways by which these smaller companies can be involved. Needless to say that throughout the whole value chain on these cellulosic ethanol productions, a very key and important role is played on who's providing the feedstock. And this is usually small players, or can be small players. We're talking farmers. And this is a beautiful new business opportunity for these players. This is a locally sourced raw material. We're not talking about commodities. This is a new revenue stream for these players. So this should not be underestimated. It also creates new job opportunities and new revenue side streams, also for smaller companies.
So there's one question, perhaps, Paolo, for you. Have you been using this technology in-house for a while to make specialty chemicals and are now licensing it because of the new regulations and the new potential for profitability in ethanol?
Yeah.
So Denise is asking this question.
Yeah, thanks, Denise, for the question. No, the answer is no. When we first started, we looked into, yes, we wanted to create a platform, but we looked into ethanol because ethanol was a commodity market, very big volumes, regulated by legislation. So that was a natural fit. So from the very beginning, that was the idea. It's actually now that we already are deploying this at commercial scale, and we sold the 5 licenses for ethanol for transportation use, that we are focusing, I would say, more and more also on biochemical and the sugar platform to differentiate the users of these products.
Okay, thank you very much. I think we've been using a lot of time. John, do you have a burning question that you would like to ask?
No, I'm conscious of the time as well.
Time, yes. So thank you very much, all. I'm going to just share with you a screen just for the last time. Whoops. There we go. And so thank you very much to all 3 of the panelists today. It was a great talk. Thank you very much. And as I said before, this was our last series of our 3 webinars. I think it was great. We had lots of different speakers from lots of different areas as well. And I think the discussions worked really well as well too. And thank you all for attending this last one, but also perhaps for the first one and the second one. All the 3 webinars have been recorded, and you can get to them either on the TOPCAPI or the CHASSY website, but also in this mcf2021.eu site as well too. So please share this with any of your other colleagues, anyone who would be interested. And again, thank you very much for the presentation, and thank you for attending the webinar.